Welcome back to the fifth and final video for Chapter 14. We'll start with Section 14.6, testing the effectiveness of different antimicrobial drugs. Testing antimicrobial drugs allows us to determine the drug's susceptibility of pathogens. For instance, can that drug actually kill the microbe? Helps us identify the most effective treatment, so which drug is the best or has the best effect on that microbe, as well as the proper dosage, what concentration, for example, should be administered. And all of this is part of routine antimicrobial susceptibility testing that happens in clinical labs. Our first test is the disc diffusion test, also sometimes called the Kirby-Bauer test, and we've done this in lab already. We've also talked about the disc diffusion test in previous video. Here we take a Mueller-Hinton agar plate that is a special formulation of the agar where it allows these different antibiotics to diffuse easily across the plate. We're going to inoculate the plate with a microbe, and you can see that as cloudy growth here, and then place paper discs soaked in different antibiotics across the plate. So each of these discs contains a different antibiotic. We'll incubate the plate and eventually see clear zones of inhibition, as we see here and here, where growth has been prevented. The edge of the growth, the junction between the zone of inhibition and where you do see growth, so the edge of that circle or zone of inhibition is the MIC, the minimum inhibitory concentration. So that's the lowest concentration of the drug as it diffuses across that inhibits growth of the microbe. To interpret the Kirby-Bauer test or disc diffusion test, what we do is measure the diameter in millimeters of each inhibition zone. So millimeters is on this side of the ruler, where if I go from here to here, that would be 10 millimeters. 10 millimeters is equal to 1 centimeter, which I see on my ruler here. So if I were to measure the diameter of this one, for instance, I would go from left to right. That would be my diameter. And then I would compare this with a standard reference chart to see if the bacterium is susceptible, intermediate, or resistant to the antibiotic. But you have to be careful because just because of a zone of inhibition is large does not necessarily mean it's more effective than another antibiotic. You also have to consider the antibiotic itself. Is it so large that it could not diffuse across that Mueller-Hinton agar in the first place? So factors that you have to take into account when measuring these different zones of inhibition include the drug diffusion rate and solubility in the agar. For instance, some drugs are very large and they do not diffuse across the medium very quickly. Agar thickness can also affect drug diffusion rate, as well as the concentration of the drug that you had in the disc in the first place. So the concentrations could be different across two different discs. The greater the concentration difference between the drug where the filter is versus over here somewhere else, the greater the diffusion rate. We also have to consider the growth rate of bacteria. So there are many variables involved. So instead of just taking by sight that one large zone of inhibition is more effective than the next kind of more intermediate or moderate size of a zone of inhibition, you usually have to use a standardized chart that takes all of these factors into account. How does one use the information from a Kirby-Bauer test or assay to predict the therapeutic effectiveness of an antimicrobial drug in a patient? So the purpose of the test was to measure how well an antimicrobial drug inhibits bacterial growth on an agar plate. We said that each paper disc is infused with some specific antibiotic and placed on a plate inoculated with the patient's bacterial culture or isolate. Then we have to interpret that zone of inhibition. So after incubation, we see that clear zone forming around each disc where the bacteria cannot grow. That is the zone of inhibition, and the diameter of the zone was measured in millimeters. We are going to compare this to a reference or standardized chart like the one we have in our lab supplement. And to determine susceptibility, we either categorize the organism as susceptible, that means the drug is likely to be effective, intermediate, where the drug might be effective at higher doses or in certain body sites, or resistant, where the drug is ineffective against that microbe. And we can also predict therapeutic effectiveness. So if the bacterial isolate from the patient is susceptible, then the antibiotic is expected to reach high inhibitory levels in the patient's tissues or blood, meaning that it should, for instance, successfully treat the infection. Whereas if we see resistance, the drug will likely fail clinically, even if given at a normal dose. So physicians will use these results to select the most appropriate antibiotic for the treatment. An antibiogram is a summary report showing how bacterial isolates from a hospital, clinic, or region are responding to different antibiotics. And it lists each bacterial species, such as E. coli or S. aureus, and the percentage of those bacteria that are susceptible to each drug. For example, if I pull up an antibiogram for E. coli, I might see that 15% of E. coli is susceptible to penicillin, but 92% of E. coli is susceptible to ciprofloxacin. These really help reduce, these antibiograms reduce inappropriate prescriptions and reduce resistance risk. So this allows us to start treatment before we get more or further confirmation from lab tests. Then we can adjust the prescription if necessary once this data becomes available. We also have tests called dilution tests, and these help us determine the MIC and MBC of a drug. MIC is the minimum inhibitory concentration, which is the lowest concentration of the drug that inhibits visible growth, and we use a broth to determine if there's growth or not. The MBC is the minimum bactericidal concentration, which is the lowest concentration of the drug that kills more or equal to 99.9% of the bacterium. So clinically, we usually aim for blood concentrations of the drug that are 3 to 5 times higher than the MIC, the minimum inhibitory concentration, to ensure efficacy of the drug. Here's an example of that MIC test, the minimum inhibitory concentration test. So what we do is, for instance, take several test tubes and we're going to serial dilute the drug in liquid broth. So you can see this is the lowest concentration of the drug up to the highest concentration of the drug. And then we add equal amounts of bacteria to each of the different drug dilutions and we incubate them overnight. Here, after incubation, I can see these two are cloudy, so those are not good at inhibiting growth, but these 3 tubes are clear. So the lowest concentration of the drug that inhibits growth, in this case, in this particular example, is 8 micrograms per milliliter. I can then use my results from that minimum inhibitory concentration test. Remember, I had clear tubes starting from 8, also then 16, and in my other example, it was 32 micrograms per mil. And I'm going to take a loopful of each of these to inoculate onto my agar plates in order to determine the minimum bactericidal concentration of the drug. So when I do that, I can actually see growth because for turbid tests, turbidity or cloudiness tests, it's hard to tell that there are actually no microbes in there. There could be just very few, which makes the tube kind of look clear. Once I do my testing on the plate, I can look at whether colonies are present or not. And in this case, the minimum or lowest concentration that was bactericidal is this one right here, because the 16 microgram per mil tube is the one that shows no growth at all. And just a quick recap, remember the minimum inhibitory concentration tells us that the drug is bacteriostatic, right? Bacteriostatic. We're inhibiting growth, but the MBC, the minimum bactericidal concentration, tells us if the drug is bactericidal, right? The lowest concentration to kill, whereas this is the lowest concentration to inhibit growth. We can also use a microdilution tray and test several different types of antimicrobial drugs in a single test at the same time. So in this case, they're testing, for example, clindamycin from low to high drug concentration, and they see growth. I can see a little dot that represents growth throughout all of those wells. So even at a high concentration, there's not enough to inhibit growth. It looks like this last concentration was 32, so they're saying that the minimum inhibitory concentration You have to go further. It's going to be something more than 32 micrograms per mil. For penicillin, I can see the first well that looks clear is the one they've circled here. So that's the minimum inhibitory concentration. For erythromycin, it looks like they've circled this one. That's the first well that is clear. Here's another example of the minimum inhibitory concentration in these wells, and in the book it marks these are clear, whereas these are turbid or cloudy, but sometimes it's hard to interpret visually just by the naked eye, so sometimes we'll be using spectrophotometers or similar devices to detect turbidity more accurately. An E-test is interesting because it combines the disc diffusion test and the dilution test into one test using a plastic strip that has a concentration gradient of your drug of interest. So this is high concentration all the way to the lowest concentration they're testing. And instead of creating a nice round zone of inhibition, it creates a zone of inhibition that has this elliptical shape. So here's my elliptical shape. The minimum inhibitory concentration is where the ellipse intersects with the strip. And here it looks like about 1.5. Yep, that's what they say, about 1.5 microgram per mil. But unfortunately, we cannot determine the minimum bactericidal concentration using this test. That previous one was kind of hard to see, so I wanted to show you what it actually looks like on the plate. And again, it combines that disc diffusion or Kirby-Bauer test with those dilution tests. Here I can see the ellipse a little bit more clearly. I can see the background bacterial growth and where it's being inhibited. Our next checkpoint again asks us to review and compare and contrast minimum inhibitory concentration versus minimum bactericidal concentration. So the MIC is the lowest concentration of a drug that inhibits bacterial growth, and we determine that using a dilution test. This tells us how much of the drug is needed to stop bacterial replication or growth, but not necessarily to kill the bacteria. That means that the culture might still have living cells, but they're not growing. And this is used to guide dosing. A lower MIC means the organism is more sensitive to the drug. The MBC is the minimum bactericidal concentration, which is the lowest concentration of the drug that kills 99.9% of the original bacterial population. And what we do is we take the non-turbid or those clear MIC tubes and we put them onto drug-free plates to see if the bacteria regrow. If no growth occurs, that concentration is the minimum bactericidal concentration. And we use this to assess whether the drug is bactericidal versus bacteriostatic. All right, let's recap 14.6. The first learning objective asks us to describe how the Kirby-Bauer disc diffusion test determines the susceptibility of a microbe to an antibacterial drug. So in this method, discs are soaked with known concentrations of antibiotics and placed on the agar plate uniformly, and the plate is inoculated with our bacterium. As the antibiotic diffuses outward, its concentration decreases with distance from the disc, and then after incubation, we see that zone of inhibition, the area with no visible bacterial growth. The diameter of this zone is going to be measured and compared to a standardized reference chart, and we'll be able to determine if the microbe is susceptible, intermediate, or resistant to that drug. This really helps clinicians select the most effective antibiotic for treating that infection. For the second one, explain the significance of the minimal inhibitory concentration, or MIC, and the minimum bactericidal concentration, or the MBC, relative to the effectiveness of an antibacterial drug or antimicrobial drug. The MIC is the lowest concentration of drug that inhibits visible bacterial growth, and that shows the drug's ability to stop replication. Whereas the MBC is the lowest concentration that kills most 99.9% of the bacteria, showing the drug's ability to kill rather than just inhibit the microbe. So again, the MIC is really looking at bacteriostatic drugs, and because we're looking at whether it inhibits the growth, but doesn't necessarily kill the microbe. Whereas the MBC tells us if the drug is bactericidal, it kills the bacteria directly. So clinically, these values really guide dosage and the drug choice to ensure that concentrations in the body are high enough to inhibit or kill the pathogen effectively. In our last section, 14.7, we're going to look at strategies for discovering new antimicrobial drugs. So because of rising antimicrobial resistance and our superbugs, some pathogens are even now pan-resistant, which means there are no effective antibiotics. We really need to find new drugs, either new natural drugs, man-made, or hybrid compounds. So scientists are exploring both traditional and more modern discovery methods. One approach, for instance, is semi-synthetic modification, where we modify drugs for improved activity. We also have high throughput screening, which is when we screen thousands of chemical compounds to see if they can be antimicrobial. iChip technology allows us to grow within the original location where the bacteria grow unculturable or previously unculturable soil microbes. And we also have combinatorial chemistry, which allows us to synthesize or produce large sets of different chemical variants of drugs to see if they have antimicrobial activity. A breakthrough tool called iChip allowed scientists to grow previously unculturable soil microbes in their natural environments. So there are many microbes that we cannot grow at all outside of where they typically live in their natural environment. But again, this helped us overcome that. And through this method, researchers discovered Texobactin, and this came from Turkish soil. This is an antibiotic that targets cell wall synthesis in gram-positive bacteria, and pretty remarkably, no resistance has been observed so far. We also have to consider new potential sources of antimicrobials, including the soil, like I just mentioned. Marine environments are largely unexplored. Microbiomes, such as the human microbiome, or synthetic biology, where engineered microbes can produce novel compounds for us. Next generation strategies also focus on resistance inhibitors, like beta-lactamase blockers that might restore the function of older antimicrobial drugs. For instance, we talked about using clavulanic acid along with amoxicillin to prevent resistance. Another next generation strategy is to target virulence factors. These are factors that pathogens release to cause disease in the body. So we have these anti-virulence therapies which disarm the pathogen, making them unable to make us sick, and we're not killing them, but that reduces the selective pressure for resistance and allows our immune system to come in and clear the infection. So new sources and strategies for developing drugs to fight infectious diseases include a focus on uncultured or previously uncultured microbes, genome-guided drug discovery, synthetic modification, or new or novel biological strategies such as using that phage therapy, bacteriophage that can kill bacteria. Phage don't kill us, so they don't harm us. So these approaches aim to overcome resistance and really expand the antibiotic pipeline beyond the traditional soil-derived compounds we have. In general, we have great technology in medicine, so why do we have so few new antibiotics? One of the key issues is that it's not very profitable and it's expensive for pharmaceutical companies to develop antibiotics. Antibiotics are short course therapies, right? Patients don't take them forever. They take them for a short period of time and not all of the time, unlike chronic disease drugs, which are really the money makers for these pharmaceutical companies. So from the 80s to about 2012, approvals of antimicrobial drugs really dropped dramatically. But luckily, in more recent years, that's not mentioned in the book, because of mostly private funding, but also some public pressure, there has been some supported renewed antibiotic discovery efforts that's led to a modest upward trend in approvals. So here was the trend we saw in the book, where there were fewer and fewer new antimicrobials approved by the FDA. Luckily, a more recent trend has shown an increase in the number of approvals, and largely this is privately driven. So we really need more proposals for public and private partnerships to share research and discovery costs. And there's a lot of ethical debates behind this. Should drug discovery be profit-driven or publicly funded? The overall goal is that we need to ensure we have a continuous pipeline of new antimicrobial options. All right, let's recap 14.7. Describe the methods and strategies used for discovery of new antimicrobial agents. So modern antimicrobial discovery uses both traditional natural product screening as well as advanced technological approaches to find new compounds. So one of this involves rediscovery and modification of natural products. Many antibiotics still come from soil microbes, especially that group Actinobacteria. And researchers can now use metagenomics and unculturable microorganism recovery tools like the iChip, which enables microbes to grow in natural environments. We also have synthetic and semi-synthetic chemistry, where we have chemical modification of known antibiotics in order to overcome resistance and improve their pharmacological properties. We have combinatorial chemistry and high throughput screening, which means we're using these automated systems to test thousands of different chemical variants to see if they're effective against these different bacteria. We also have genomics and bioinformatic approaches, which allows us to scan or screen the genome to identify silent or other types of gene clusters and microbes that might help us find or identify new antimicrobial compounds. And then we also have repurposing studies that take non-antibiotic drugs or combinations of drugs and test them for antibacterial effects, or we can even use these to boost existing antimicrobial efficacy. All right, and that takes us to the end of chapter 14. In the next chapter, chapter 15, we're going to look at microbial mechanisms of pathogenicity. This is how pathogens cause disease. Thank you for watching.